Okay, biologists, I'm just going to run through your nucleic acids, proteins, and gene regulation paper to help you to build your error log. So take particular note of any areas where you've dropped some marks and ensure that you've got detailed notes addressing that. Close any learning gaps and make sure you don't make the same mistake again. Just running through section B because the multi-choice should be fairly straightforward. Let's have a look at question 1. Now this table was almost a direct copy of the one that you've done in your workbook on page 18. It will become easier as you move through Unit 3 to identify the different functions of proteins. One thing I want to draw your attention to is hormones. Taking you back to Year 9, hormones are chemical signals released from endocrine glands, and testosterone and estrogen are not protein hormones, they're cholesterol-based hormones, and they would not have been given a mark at all. Okay, I've given you some examples here on the paper that you could have used, so correct that one, and note that you cannot use any hormones that are cholesterol-based. They need to be amino acid monomers built into a polypeptide chain, as every protein is. What is the proteome? It was important that you'd actually watch the video that I'd set, and when you describe proteome, you talk about the total amount of proteins produced by not only an organism but also a tissue, so a collection of cells, or a single cell. We can look at the proteome of any number of things. Saying that it's the total amount of proteins from an organism would not have been enough. Then you need to cite some specific applications for proteomics. One example of that might be investigating medical conditions, developing different types of drugs. You need biotechnology in general, but possibly a little bit too general. Please do a little bit of research around this if you're not clear on what the proteome is, and come and see me. Question 2 was interesting. There was a little bit of text here, and not many of you actually annotated that text. It's important to use the terms that the question used in your answer. So I wanted you to refer to ricin when appropriate, okay? And really understand that ricin is a toxin and it's a protein, and this is the structure of that protein, and show your understanding of those ideas in your answer. A is direct recall. So these are the easiest questions to get marks on. What level of structure is indicated? Secondary, identify X and Y, alpha helix and beta pleated sheet. If you're not clear on the levels of protein structure and how they fit together, revise that and come and see me if you have any problems. Most people dropped marks, if at all. They dropped marks in this section 4 at the bottom. I want you to annotate these questions and actually look for what the question is asking you to give. So what level of structure will this change? A change in the sequence, so they're talking about the DNA sequence, will affect the polypeptide chain, potentially, and that will impact its tertiary structure. Okay, so if you change the R groups, you change the interactions, and you change the three-dimensional shape of that protein. And then notice it wants to know how this will affect the overall function of the protein. So it's not enough to just say the protein function may be reduced. You need to tell me that you know what the protein function is. Therefore, the toxicity of ricin would be reduced in all likelihood. So I was looking for those terms specifically. You need to link your answers back to the question and the terms in the question. Okay. B here talks about inactivation of ribosomes. To achieve both marks there, you need to be able to tell me that ribosomes are the site of protein synthesis. And if ribosomes are inactivated or turned off, proteins won't be made. And why are proteins involved in functioning of a cell? Well, because proteins are involved in most biochemical reactions, as you know. Therefore, compromising, if you knock out some of those proteins, you compromise many, if not all, of the cell's functions. Question 3, giveaway mark pre-mRNA. Question 4 here It was a skills test to see if you could read the codon table. So know that these are always codons, even if it doesn't tell you. They're clearly not DNA sequences because there are uracils in there, and it's not tRNA, it's messenger RNA codons. I would like you in these formative tasks, so everything is formative until November. In these formative tasks, show me your working and your thinking so that I can follow things through. So I wanted to see that you were turning the DNA sequence into messenger RNA, and then you could read the table and identify the correct amino acid chain. Moving on to this one. So here we're looking at question 5. I wanted you to annotate this question. So 5a, the process shown by X produces a copy of the DNA molecule and circle, underline this bit, and modifies the product. So it's copying the DNA, building messenger RNA, and then mRNA modification is occurring, okay, to allow it to exit from the nucleus. You know that messenger RNA pre-mRNA cannot leave the nucleus until introns are removed and it has been capped. Many people missed that. Consider the key terms that you're looking for here, and the activity we did directly before the test should help you. Outline the steps. I wanted RNA polymerase, the promoter region, the template strand, adding free nucleotides to build a pre-messenger RNA molecule using the reaction condensation polymerization. This occurs until the termination region is reached, and then RNA polymerase will detach. The messenger RNA is modified with introns removed, sometimes exons also removed, which is the alternative splicing, and the methyl cap being added to the 5' and the poly A tail added to the 3' end. That's what I was looking for. If you are missing details, please add them and write yourself the perfect answer. Here, what advantage would being able to switch off genes confer to a cell? Notice this is a 2-mark question, so save energy is not enough. You're saving energy and saving resources. And why are you saving energy and resources? You're saving them, or how? You're saving them because you're only making proteins when they're actually needed. So those were the 2 points here. Conserves energy and resources, 1 mark, by the how, producing proteins when needed. Over on question, gene regulation. Describe the events that lead to the prevention of the production of the enzymes that break down lactose. Point 1, the prevention of production of enzymes will occur if lactose is not present. That's your first mark. You can't jump into it without describing the environmental situation that will turn off these proteins or the synthesis of these proteins. Then repressor will bind to the operator region. Really important that you state repressor protein and operator region. RNA polymerase therefore cannot transcribe the structural genes. And therefore gene expression is prevented and the enzymes will not be produced. They're the 4 marks. Please improve your answer. Notice which key terms you're missing. And the final page, this one, so we're still on gene regulation. Identify the difference between structural genes and regulatory genes. This is a whereas question. Start with regulatory genes. Describe what regulatory genes do, and then explain how structural genes differ. So regulatory genes produce a molecule that controls gene regulation, whereas structural genes produce proteins or molecules that have no relationship or do not control gene regulation. Okay, you needed both of those statements to get both marks. Repressor protein is a protein that binds to the operator region and can turn off gene expression. Straightforward recall. And the operator, it doesn't say the operator region, but the operator is a regulatory sequence. So it's still a DNA sequence, but it does not lead to a protein being produced. It doesn't code. It's not a gene as such. It's what we call a regulatory sequence. and it's the place where the repressor protein binds in the operon. Okay, I hope that is useful, and please come and see me if any of this doesn't make sense or you want any support in closing those gaps. Well done.